Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course on computer aided drug design. In this class we are going to look at drug properties. So it is not only that a drug should have very high activity, whether killing bacteria or whether killing cancerous cells or whether reducing the glucose levels in the blood, it should have a large number of important properties. I did introduce some of those properties, but we will start going more in detail. Okay, so because it is taken inside the human body, okay, it is consumed by all of us mostly orally, because a large number of drugs are consumed orally when compared to intravenous or intraperitoneal. So the drugs should have activity, of course, it should have good activity, and it should have a selectivity for single target. We do not want drug acting on several targets, thereby it may be causing side effects. So it should be very specific to one particular target. So if it has to kill a particular type of tumor, it should go and attack only that. If it has to go and block an enzyme in certain pathway, it should go and block only that enzyme, not all other enzymes in that pathway. Then it should have good oral absorption, because as I said, most of the time we take drugs orally because nobody likes injections and different modes of intake of drugs, and it should get distributed inside the body, because as soon as it reaches the blood, the drug has to get distributed everywhere, and then it will go and reach the target site, and of course it gets metabolized inside, okay, in the body. So let us look at this particular figure which I have drawn. So generally we take a drug orally, so it reaches the stomach, okay, there is an absorption in the stomach, then as it goes down in the duodenum there is absorption inside into the bloodstream. Of course the liver is there, which starts degrading because liver is a wonderful gatekeeper. So there are many enzymes, P450 type of enzymes, okay, they degrade, the drug gets metabolized. So only small amount of drug is available in the bloodstream when compared to what the drug we take here. So if the drug does not get absorbed in the GI tract or in the duodenum, it gets excreted, it is a waste, okay. And again inside, if there is a metabolism, and then it goes liver to kidney, and then drug again gets excreted through urine, again that is a waste. So what is available in the plasma could be much less than what is taken in orally. And of course it does not stop there, some of the drug can even get absorbed in the tissues. So the amount of drug that reaches the target site where it has to go and act may be much less when compared to drug which we take here, okay. So this is the effective concentration where it acts, whereas this is what we take, okay. So this ratio is also called bioavailability. And so imagine I have a swelling in my finger, and we all know we take drugs like ibuprofen, they are called non-steroidal anti-inflammatory drug. So it comes in, whatever gets absorbed goes into the bloodstream, but whatever does not get absorbed into the GI and duodenum is wasted, and the liver starts degrading the ibuprofen, so it may come out through urine, again it is excreted, and there could be some absorption from the bloodstream into the tissues. So the concentration that reaches my finger to reduce the swelling in my finger may be much less when compared to what I take orally, okay, because this is the concentration which acts on the target site, and the action will depend upon this concentration and not this concentration. So a lot of things happen for the drug when it moves from your oral to the target site, okay, things like absorption, distribution, metabolism, because it gets metabolized inside the body, enzymes like oxidoreductase, lipases, okay, and so on, actually esterases and so on. Okay, then of course it gets excreted, which may not get absorbed in the stomach or the duodenum, or it can get excreted from the kidney through urine. Then solubility, as you know, the pH of the stomach is 2, whereas the pH down the line can go up 2, 3, 4 and all that, and the pH in the bloodstream is 7.4. So it has to be soluble at all these pHs. So that is a big challenge for the drug. It should be soluble at 2 or it should be soluble at 7, otherwise it will start precipitating. Okay, some of the natural products might not be soluble here, they may be soluble here and so on, actually. Okay, so many of the drugs are made into salts so that it improves the solubility here. And then this bioavailability, that means the concentration of the drug at the target as against the concentration that is given orally. Okay, that is called bioavailability. If the bioavailability is large, so even with small concentration we achieve reasonably good concentration here. If the bioavailability is poor, even if you take very large concentration of the drug, the concentration available at the target site may be very, very low. For example, I have a fungal infection in my foot. Okay, so the concentration at that foot should be higher so that it kills the fungus. Okay, so I take an antibiotic, but the antibiotic goes through all these processes. Some of them gets absorbed, some of them gets excreted, then some of them get metabolized in the liver, goes away as urine, some of them might get absorbed in my tissues, and so the concentration that is reaching my foot, if it is less than the minimum inhibitory concentration required to kill the fungus, whatever I have taken is of no use. So the concentration of the drug at my foot should be higher so that the fungus get killed, otherwise doctor has to prescribe much higher dose, which can lead to toxicity in the stomach or liver, but it may not act at the target site because it is not reaching the minimum inhibitory concentration. So the bioavailability of a drug should be high. Generally they say about 30-35% at least you should have a bioavailability. That means 67% of the drug is wasted somewhere. Okay, either as excreted, either as tissue deposits, either as metabolized. Too much metabolism taking place inside the body because some of the structure features are so bad that it gets either oxidized or it gets esterified and so on. Then no side effects, that means the drug should not have any side effects, short or long term. Okay, so we do not want either short term, that means within a few months maybe vomiting or increase in blood pressure, or long term it should not give cardiovascular problems or some other side effects in the long term. Okay, then no toxicity, of course it should not have any toxicity. Okay, so there are 10 points which we saw and it is not complete, you are going to see some more of them. Okay, metabolites also should not be toxic because the drug may not be toxic, but it degrades into smaller molecules which may be toxic. So we do not want, the drugs should be stable, just like soluble at these 2 pH conditions, it should also be stable at these pH conditions. Okay, there are, suppose there are ester bonds or amide bonds, they may start degrading or getting hydrolyzed at acidic pH. Okay, then there is something called blood brain barrier, we will talk about this later quite a lot. This blood brain barrier separates the entire body with the brain. Okay, it prevents viruses, other toxin entering the brain, it allows only oxygen, nutrients to pass through. Okay, so the drugs which we take for curing the human body should not enter the brain and start disturbing the brain function. Okay, so there is something called a blood brain barrier, we do not want the drug to penetrate that blood brain barrier. Okay, we do not want ibuprofen penetrating and causing some issues with the brain. Of course there are drugs which are meant for neuro diseases, okay, central neural system related diseases. At that time we want the drugs to cross the blood brain barrier, whereas normal drugs which are used for our body functions should not be crossing the blood brain barrier. We will look at that more in detail later. Okay. And there is set of P glycoprotein type of proteins which acts as a effluxing pumps. That means if they see any foreign molecule, they capture it and throw it away. Okay. So that way they keep the body free of toxins, especially in the blood brain barrier region, especially the pregnant women, they all have quite a lot of this. So we do not want the drug to get effluxed out, then it becomes a waste. So effluxing happens, the drug will get thrown out into the urine. So we do not want that to happen, because we lose drug, whatever is present inside the plasma. Then shelf life, of course you make the drug, the manufacturer makes it, and then it is sent to pharmacy and then the patient buys it. So it may take 3 months, 6 months. So the drug should have enough shelf life, that is very important. It should not start degrading, it should not get oxidized and so on. And you have to decide on the form. Is it going to be a oral drug, it is going to be intravenous, intraperitoneal, intramuscular, nasal, anal, vaginal, dermal. So depending upon the type of drug intake, they may formulate it in different ways. Okay. So that is also very important. Okay. It should be easily made into a powder form, or it should be made into a solution form, and so on actually. And we should also know the mechanism of action, should know how the drug acts, which enzymes it blocks, or which proteins or which genes it down regulates or up regulates. That is very important. Nowadays that question is being asked by the FDA, that is the Food and Drug Administration of USA, before giving approval. They do not just give approval for any molecule which shows very good activity. It was so many years back, but in the past 25-30 years, without knowing the mechanism of action, drugs are not approved by FDA. And of course cost plays a very important role. If it is a new drug, new chemical entity introduced, so pharma companies will charge very high prices to recover the R&D cost. Such drugs are not sold in countries like India or third world countries, but once the drug comes out of the patent, okay, once the drug comes out of the patent, then it becomes a generic drug, then the cost comes down. Okay, some of the drugs, for example, for tuberculosis, HIV, malaria, generally it is expected the cost of the drug has to be low, because it has to reach out to very poor population. Whereas some of the cancer drugs which are being introduced very newly may cost thousands and thousands of rupees, okay, whereas a malarial drug will cost only few rupees. So then ease of manufacture, of course bioprocess, how do you manufacture this, is it easy to manufacture, that is also very important. You should be able to manufacture it in very simple steps. So and then ease of animal trials, because once the new chemical entity is established in the lab, the trials have to be done on animals, then with human volunteers. So it should be easy to perform these trials and monitor the entire operation. So you see almost 20 requirements are there. So it is not just activity, but a large number of requirements are needed, so that you can be very sure that it can pass out as a drug. So many active molecules in your lab do not cross the approval, because they have one of these problems. Okay, so they may be having one of these problems here, or they may be having one of the problems listed out in this. So the drug gets thrown out. So the chemistry plays a very important role when you design drugs. The chemical structure plays a very important role in deciding on the property. So the structure property relationship is very important. Which structural features improve its stability, which structural features reduces metabolism, which structural features improve bioavailability, which structural features improve solubility in the GI. Okay. So, all these are very important to know. So, you should have good knowledge about medicinal chemistry, biochemistry, physical chemistry and so on actually. Okay, so, what are these structural features? Molecular weight, hydrogen bond donors, hydrogen bond acceptors. Hydrogen bond donors means groups like OH or NH, hydrogen bond acceptors means oxygen, nitrogen, like that. Log P is a very important property. Log P tells you the hydrophobic hydrophilic balance of the drug. That means it should neither be hydrophobic or it should neither be hydrophilic. If it is too much hydrophilic, it will dissolve nicely in the stomach, but it has to cross the GI, which is made up of lipids, which is hydrophobic, so it will not cross. If it is too much hydrophobic, the drug will not dissolve in your stomach fluids, but it will be able to easily diffuse through the GI, because GI is very hydrophobic. Okay, that is called log P. So, log P is a ratio of the hydrophobic to hydrophilic balance. Then comes pKa, pKb, that means a dissociation in mild acids and mild base conditions. What is the polar surface area? Okay, polar groups are like oxygen and nitrogen. How many heterocycles are there? That means groups which cyclic groups which contains nitrogen and oxygen. How many aromatic rings are there? How many ketonic groups are there? How many rotatable bonds? Once you have rotatable bonds, drug is very flexible, so it can easily fit into an active site. Crystalline nature, whether it is crystalline or not. Too much crystallinity, the drug will not be soluble. If it is amorphous, it is soluble. How many ester bonds are there? Amide bonds are there, because ester bonds can easily degrade with esterases, amidases. If these enzymes are present in the body, they can get metabolized. Neutral or salt form, is it a neutral or salt form? So, all these features, and these are the activity, selectivity, oral absorption, distribution inside the body, metabolism, excretion, solubility at pH 2 and 7, bioavailability, side effects, toxicity, stability, blood brain barrier, PGB efflux, form. So, all these are decided by the structural features. Okay. We also call it structural descriptors. We call it structural descriptors or structural features and so on. So, we call it features, descriptors, okay, sorry, descriptors, features, structural descriptors. Okay. So, these structural features or descriptors determine various properties of the drug. So, as a chemist, medicinal chemist, you should be able to play with these to arrive at these, and of course, activity is the most important, but you cannot forget activity is the most important, but you cannot forget all these. These are all very important for drug to cross the FDA approval. Okay. So, these structural features, structural descriptors determine all these parameters. Okay. So, what happens? Sometimes we need to take a balance between activity or potency or selectivity as against the other drug likeness property, whether it crosses these GI tract, toxicity, metabolism, pharmacokinetics. Okay, what happens? So, we need to balance between activity versus other properties. Okay. So, sometimes you will have a very active drug, but it will have very poor properties. Maybe it does not cross the GI tract, or maybe it gets degraded, whereas the second most active drug in your lab may have all the other properties. So, it is better to take the second most drug into the market through the clinical trials rather than bet on the first drug, or make modifications to the structural features of the drug. Okay. Make some modifications of the structural features, okay, so that we get better if there are any issues on one of those properties. Okay, let us now look at some of the drugs which I mentioned, commercial drugs in the market, which I had mentioned as a few top selling drugs and we look at some of those structural features. Okay. This drug is called Zelzanx. This is for rheumatoid arthritis. This is taken as an oral drug. Okay, so the volume of distribution is 87 liters. That means it gets distributed in the blood as well as in some tissues. So the when you have a very large volume of distribution inside the body, the concentration of the drug goes down. So that means effective concentration is much less. Okay, it binds to a protein. The drug binds to protein by about is about 40%. Okay, and so its bioavailability is about 74%. That means if I take 100 milligram of the drug orally, 74 milligrams of the drug is available at the target site, which is good. Generally things like 70, 75, 65 are reasonably good. Okay. It gets eliminated from the body. The half life of elimination is 3 hours. Okay. That means 50% of the drug gets eliminated from the plasma in 3 hours, which is also good. We do not want too much of the drug staying into the body for a very long time, and of course the elimination is very fast. The drug action goes down very fast, so you may have to keep taking the drug quite often. If the elimination is very slow, then there is an accumulation of the drug, there could be other problems as you go along. Okay. Okay. So metabolism, drug gets metabolized by this particular enzyme called cytochrome P450. Okay. So a large number of enzymes metabolize that. Okay. Okay. This is one of the top selling drugs in the market. Okay. So clearance is 70%. So it gets cleared through the hepatic clearance, okay, through the liver, and this is through the renal clearance. Okay. Let us look at another one. It is called etanercept. Okay. This is a biological product. As you can see, it's a protein. Okay. It treats autoimmune diseases by interfering with TNF, a soluble inflammatory cytokine, to decrease the signs of rheumatoid arthritis. Okay, so it treats autoimmune disease, and it is a large molecule, it is a biomolecule. Okay, as you can see, 51,234 Dalton, it has got 934 amino acids in it. So obviously it is given as an injection under the skin. So as you can see, if the molecule is smaller in molecular weight, like in the previous case, it can be given oral, whereas this is a very large molecule. Obviously proteins cannot be taken in orally, because when the pH of 2 in the GI, the stomach, it may get degraded and it will also not cross the GI tract. So its half life is quite large, 102 hours. Okay. Its clearance is 60 to 80 milliliter per hour. Its isoelectric point, that is 7.89, that pH at 7.89, it has got 0 charge. Okay. So it is given as injection. So because it is a protein, large molecular weight. Let us look at another one. This is a chimeric monoclonal antibody that works against TNF alpha and is used to treat autoimmune diseases and rheumatoid arthritis. Again, this is a large molecule, it is a protein, it is a chimeric protein. Okay. So 144,190, okay, 144,190. So obviously it is given intravenous here. As you can see, it is given intravenous. Bioavailability is excellent, 92%, because it is given intravenous. There is no problem about crossing the GI, pH stability, all those things. Volume of distribution is 3 to 6 liters. So very low volume. So obviously concentration in the blood plasma region will be very high. Now look at another drug. Okay, this is a small molecule drug. This is a mixture of 3 drugs: Advar, Seretide, Vienna. Okay, it is a formulation containing so many different, used in the management of asthma and chronic constructive pulmonary disease. Okay, so it contains some anti-inflammatory. So it contains drug which will dilate the pulmonary they help prevent symptoms of coughing, wheezing, shortness of breath and so on. Okay. And so obviously low molecular weight 916 grams mole. Okay. So it is basically constriction of the airways, it is for anti-inflammatory. Look at another one, this is a long acting basal insulin analog. Okay. So insulin analog means it is supposed to control the blood sugar level. Okay. It is a long duration, long action, so it is 18 to 26 hours. Okay. Produced by recombinant DNA technology, it is an isoelectric point of 5.4 to 6.7, half life 30 hours in mammals, 6063. Okay. So it is not given orally, it has to be given through intravenous. Okay. Look at another drug, a monoclonal antibody against the protein CD20. So one important thing you notice that all the commercial drugs, top selling drugs which I am showing here, the mechanism of action of these are well known. Okay. It is again it is 143,859 large molecule, so obviously it is given intravenous, half life is 30 to 400 hours. Okay. Because it is a protein like, it has got 213 amino acids, 2 heavy chains, 451 amino acids and 2 light chains, 2 heavy chains, 2 light chains, 213 amino acids. Okay. This is given meant for autoimmune disease, okay, leukemias and so on actually. A volume of distribution 3 liters, half life is 0.8, clearance is very slow, 0.34 per day. So it can act much longer. Look at another drug, this is called Avastin, it is a recombinant monoclonal antibody that blocks angiogenesis by inhibiting vascular endothelial growth factor A, VEGF-A it is called. So it is involved, it prevents cancer, prevents blood vessel proliferation, okay, and tumor growth. Okay. Of course this is a recombinant product and it is given intravenous because look at the molar mass 149,196, half life is quite good, 20 days, that means it lasts inside the body for very long time, that is why clearance is also very low, 0.26, and volume of distribution is 46 ml, very low. So little concentration, I mean little dose inside, you can have large concentration. Look at another drug, trastuzumab, that is brand name, Herceptine, it is a monoclonal antibody that interferes with the HER2 new receptor to treat certain breast cancers here. Okay. Okay, it is a huge molecule, 145,531. Okay. Half life of 28 days, volume of distribution is 44 ml per kg, that means volume is very low. So you can achieve very large concentration with this drug. Okay. So this is also given intravenous. So as you can see, many of these biological molecules have very large molecular weight, and so most of them are given intravenous because of its stability at GI pH of 2, as well as it will not be able to cross the barrier. Look at this drug, rosuvastatin. Okay, this is class of statins to treat high cholesterol. Okay, it is a competitive inhibitor of HMG CO reductase. Okay, and small molecular weight 481, and look at this, it has got very low bioavailability because it is taken orally, that is why the bioavailability is very low, 20%. So if drugs are taken intravenous, you will have very high bioavailability, but nobody likes injections, everybody likes oral drug. Once you have oral, bioavailability could be one of the problem. Why is it so? It binds to a lot of proteins inside the body. Okay, it is reversible, but because of this binding, concentration of the drug in the plasma comes down. Okay, peak plasma concentration is 3 to 5 hours, volume of distribution is very large, you can see 134 liters. Many of the protein type of drugs have very low volume of distribution, whereas these type of small molecules have very high volume, so the concentration available inside could be very low. Okay. So statins are big business for reducing cholesterol there are huge number of statins, atorvastatin, serivastatin, pitavastatin, rosuvastatin and so on, but this particular molecule contains sulfur, other molecules do not contain sulfur. So, statins are meant for high cholesterol and it is a big business, annual sales could run into billions of dollars. Look at another one, that is the atorvastatin, marketed under the trade name of Lipitor. Once upon a time it was making tens of billions of dollars, okay, for reducing cholesterol by acting on this HMG-CoA reductase, okay. So, it, as you can see, 1996 to 2012 Lipitor made 125 billion sales, okay, that is a huge scale business, okay. Again the volume of distribution is 381 liters. So, obviously the concentration inside will be low because a lot of protein binding takes place and the bioavailability is also very low, 14%, and systemic availability is 30%, so it is quite low, okay. Maximum concentration is achieved in 1 to 2 hours, so it starts acting very fast, okay. Look at another drug, this is also a small molecule under the brand name Abilify, okay. This is meant for psychotic disorders, it is called CNS type of disorders, central nervous system related disease. So, it has to cross the blood brain barrier, this is a small molecule, 448 molecular weight, bioavailability is quite good, 87%, it binds quite a lot to the protein, it gets metabolized by cytochrome enzymes in the liver, okay. Biological half life is 75 hours, is excreted renal by 27, fecal by 60%, volume of distribution is reasonable. So, large number of highly successful, commercially successful drugs, structures and properties which we saw now, some of them are biomolecules, proteins, obviously they are given as intravenous, some small molecules are given orally, when they are given orally bioavailability goes down, as you can see. When they are given intravenous, bioavailability is very large. Proteins have a very low volume of distribution, so peak concentrations can be very high, whereas small molecules have very large volume of distribution, so peak concentrations could be very low, okay. So, we look at very important properties which a drug should have before it becomes successful. So, you need a lot of knowledge about biochemistry, medicinal chemistry, physical chemistry, so that you can combine all these aspects with the biological activities, so that one can come up with a very successful molecule. So, we will continue more about these drug properties as we go along, okay. Thank you very much for your time.